my name is Thomas Washington, and I am a first-year graduate student in the Genetic Counseling program at the University of Pennsylvania. In this presentation, I will be talking about MED13L genetic reports. Breaking it down further into the subtopics I will be hitting, we will first go through a genetics crash course, briefly discuss MED13L biology, and then spend more time on types of genetic variants you can see in MED13L, as well as how to read and understand a MED13L genetic report. When talking about reading MED13L genetic reports or any genetic reports, two of the topics that can bring up a lot of questions are de novo variants versus germline mosaicism and variants of uncertain significance. So I will go into a little bit more detail on these two topics. So starting with that genetics crash course to set the context for the rest of the talk, you have trillions of cells in your body. Most types of cells in your body have a nucleus, which is the part of the cell that contains chromosomes. Our DNA is packaged into these chromosomes. You can think of all of your chromosomes together as a book and each individual chromosome as a book chapter. We have genes located along chromosomes, and you can think of each gene as a paragraph. Within genes, we have codons that are like words, and those codons are made of nucleotides, which are like letters. You may have heard that genes provide instructions for making protein, and that's true. In this book analogy, the paragraph gives you information, just as a gene gives the cell information on how to make protein. I do want to go back to the codon part for a minute and note that each codon is made of three nucleotides, or letters, and the cell will always read codons as a group of three, which is important and will come back up later. Moving on to talking a bit about how MED13L, the protein, functions and what it does in the body. MED13L, the protein, is a part of the cyclin-dependent kinase module of the mediator complex. The mediator complex is a bundle of multiple proteins that is important for the creation of proteins from genes. A mouse model study suggests MED13L is involved in central nervous system development and nerve cell communication. MED13L is important for mitochondria to work as they should, and up to 90% of the energy a child needs to function and grow is produced by mitochondria. MED13L syndrome is primarily considered a haploinsufficiency developmental disorder, where haploinsufficiency means that half is not enough. For a little more context on that point about haploinsufficiency, we have two copies of each gene, and if one copy of the MED13L gene is not working as it typically should, it causes MED13L syndrome. Here we are looking at some of the ways that a copy of the MED13L gene can be made to not work as it typically should. To give some more context, the top part of each box shows the type of variant or change in the DNA, and the bottom part of each box shows what the variant does. Within that bottom part of each box, we have the sentence, "The cat saw the rat." Each word of this sentence is a codon, which code for amino acids that make up a protein. So for the first one on this slide, a small deletion in the gene causing a frame shift. This shows a frame shift variant where the letter E from the is deleted, causing a shift in the way the codons are read. This alters the subsequent words or codons and leads to a completely different sequence of words. We can see that the sentence no longer makes sense. For a small duplication or insertion in the gene causing a frame shift, this shows a frame shift variant where the letter H from the is duplicated, causing a shift in the way the codons are read. This again alters the words or codons that come after, and again, the sentence no longer makes sense. The shift in the way the codons are read occurs whenever the duplication or deletion isn't in a multiple of three. For a partial gene deletion not causing a frame shift, this shows a deletion where the words cat and saw are deleted. So here, a big chunk of that sentence is taken out, and we have lost the meaning of the sentence. For the nonsense variant, the sentence ends before it should, and then for the missense variant, we are changing one letter or word. The C in cat was changed to an X, and the sentence doesn't make sense even though the other words are unchanged. Remember that MED13L syndrome is caused by haploinsufficiency. So if you have one of your two copies of the MED13L gene not making protein or making the protein incorrectly, it can cause the condition because half of the typical amount is not enough. All of the types of variants that we looked at on the previous slide can cause this, but it happens in different ways. Frameshift variants result in the protein being made improperly, so too long or too short with many amino acids changed, or not at all. With the partial gene deletions not causing a frame shift, many codons are deleted from the gene, and a big chunk of the instructions to make the protein are missing. This can result in a loss of functional protein. Nonsense variants cause a codon to deliver the message, stop making the protein, before the protein is finished. This could result in a shortened protein, but likely results in no protein being made. In missense variants, one amino acid of the protein is changed, and the protein may or may not function as it typically does. There are some other types of variants that can cause MED13L syndrome that we haven't touched on yet, including whole gene duplications and splice site variants. In whole gene duplications, the whole gene is duplicated, resulting in 3 total copies of the MED13L gene. This is different than a small duplication within the gene causing a frame shift and is usually associated with a milder presentation. Splice site variants are changes in DNA that affects how it is processed into instructions for making protein. With the processing disrupted, the instructions are disrupted, and the protein can be made improperly or not at all. Now we are going to be getting into the part of the presentation about reading genetic reports. Here we see a result table from a genetic report. The first column is the gene column. Genes are made up of DNA and are the basic units of heredity. Humans have about 20,000 genes used to make proteins. Proteins have many roles in the body, including being enzymes, hormones, and building blocks. Mouse model studies suggest MED13L is a protein involved in brain development and cell-to-cell communication in the central nervous system. A study in human cells also found that MED13L is important for mitochondrial function. I also wanted to note that when the lab is going through and interpreting variants that they find during genetic testing, it's important for them to know the gene and know if it has previously been associated with a condition. The next column is the disease column. The name in the red box is the name the lab decided to give the condition. The same condition can have multiple names, so you might call this MED13L syndrome. The importance of having the condition or disease listed here is that one gene can have multiple conditions associated with it. At this point, it doesn't seem like that is the case for MED13L, and if you see multiple disease or condition names with MED13L, they are likely just different names and not different conditions, although this is something that can change over time with more research. The mode of inheritance for MED13L syndrome is autosomal dominant. In general, autosomal refers to the non-sex chromosomes. If you look over at the image with the chromosomes in the bottom corner of the screen, most of the chromosomes have a blue background behind them. Those are autosomal chromosomes or non-sex chromosomes. The chromosomes with the purple background are the sex chromosomes and determine male or female sex. With this condition in this gene, the genetic variant is on one of the non-sex chromosomes. Dominant means that one altered copy of the gene is enough to cause the condition. Recessive means that both copies of the gene must be altered to cause the condition. An example of a recessive condition is cystic fibrosis. Here I'm going to be focusing on the variant column, but just the C dot portion that you can see in the red box. The C dot means that we are talking about the coding DNA. So here, the 4,987th through 4,991st nucleotides, or the letters that make up DNA, of the coding DNA for this gene were deleted. The INS stands for insertion, and the 16 in parentheses after INS means that 16 nucleotides were inserted. So, with 5 nucleotides deleted and 16 inserted, that would equal a total change of an extra 11 nucleotides. Because 11 is not a multiple of 3, this results in a frameshift variant. Other frameshift variants may have just INS or just DEL instead of both like we see here, but the overall effect is the same. I wanted to go back to this slide from earlier to highlight which type of variant we see in this report table. It's really a combination of the two types boxed in red because some nucleotides are deleted and some are inserted, with the result being a frameshift. Now for the P dot portion of the variant column. The P dot means we are talking about the protein, MED13L. T refers to the amino acid threonine, sometimes written as THR. 1663 is an amino acid position, so we are at the 1663rd amino acid in MED13L. C refers to the amino acid cysteine, sometimes written as CYS, The FS means frame shift. The star 43 means that the frame shift variant is causing the protein to end 43 amino acids after amino acid position 1663, which is not where it would typically end. Altogether, the P dot is saying that threonine is typically the 1663rd amino acid in MED13L, but this variant changes it to a cysteine. The variant also causes a frame shift with the protein ending 43 amino acids later. As we talked about earlier, frame shift variants result in proteins being made improperly or not at all. For the zygosity column, we have heterozygous, which means that the variant is present in one of the two copies of the gene. In the inherited from column, it says de novo. This means that the variant is a new change in this individual, so it was not inherited from either parent. I wanted to talk more about de novo variants in germline mosaicism because it can be confusing and it is an important topic. If a lab is reporting out a de novo variant like this, they will probably have a statement like the one you see here highlighted on the slide. As both parents tested negative for the variant, most likely it arose de novo in the child. However, the possibility of germline mosaicism must be considered. The top image here is showing the scenario of a de novo variant. Gray means no variant, and orange means there is a variant in MED13L. So, neither parent has the variant, and the child does. This could be due to a variant arising early in the child's development when they are still an embryo. The bottom image shows the scenario of germline mosaicism. The sperm contributor is not affected by MED13L syndrome because most of the cells in their body do not have a MED13L variant. However, they do have cells in their testes that have a MED13L variant, and so they do produce sperm with a MED13L variant. This sperm can can combine with an egg and result in a child with a MED13L variant. If a child has MED13L syndrome due to a de novo variant, the chances that the child's parents have another child with MED13L syndrome are very low. If a child has MED13L syndrome due to germline mosaicism, the chances that the child's parents have another child with MED13L syndrome are higher. It can be incredibly hard to test for germline mosaicism, though, because if you do genetic testing on the parent's blood, as we normally do for genetic testing, it will come back without the variant, as most of their cells don't have the variant. Even if you try to test the testes where we suspect they may have a variant, you can't really know if you are taking from the right part of the testes. That test could come back without the MED13L variant, but you still wouldn't be sure if there are other cells in the testes that do have the MED13L variant. I also want to note that I could have shown the mom with the orange circle and orange egg. Either parent could have germline mosaicism. The last column in the result table is the classification column, which in this case is pathogenic. Everyone has many variants in their DNA. Most of these do not have any significant impact on our health or development. Therefore, the lab classifies variants or puts them into categories based on how likely they are to be disease-causing, pathogenic, or non-disease-causing, benign. The classification is done using scientific evidence. The amount of evidence and how strong or convincing that evidence is determine which category a variant is placed in. Going through these categories, pathogenic means the variant is considered disease-causing. Likely pathogenic, there is a high likelihood that the variant is disease-causing. Variant of uncertain significance, which you can see as VUS or VOUS. The variant has characteristics of being disease-causing, but there is insufficient or conflicting evidence. Likely benign, there is a high likelihood that the variant is not disease-causing. And then benign, the variant is not considered disease-causing. As I said earlier, I wanted to go into a bit more detail on variants of uncertain significance. So again, if something is classified as a VUS, the variant has characteristics of being disease-causing, but there is insufficient or conflicting evidence. A lab may be highly suspicious that a VUS is causing a condition, but there just isn't enough evidence at that time to call it pathogenic. However, evidence can change over time as more research is conducted, or maybe you see the same variant in other individuals with the same condition. Because evidence can change over time, a VUS can be reclassified to another category later, such as moving from VUS to likely pathogenic, for example. If your child does have a VUS in MED13L, the brain gene registry is something you can consider getting involved with. It is an NIH-funded research study, and MED13L is a top gene of interest. As part of the Brain Gene Registry, you have the opportunity to enroll in Genome Connect so you can get notified if your variant is being reclassified. Reclassification from VUS to pathogenic or likely pathogenic is important for several reasons, including getting a clear diagnosis of MED13L syndrome to better rule out another cause of symptoms. It helps in being included in clinical trials, and it can lead to a better understanding of symptoms in the clinical and research communities. This link at the bottom of the slide is where you can find more information. These last couple of slides show other examples of variants you could see in the variant column in a genetic report table. The first one is a missense variant, where one nucleotide or letter in the MED13L gene is changed from a C to a T, causing one amino acid of the protein to be changed from a serine to a leucine. Again, missense variants can disrupt protein function. Next is a nonsense variant, where one nucleotide or letter in the MED13L gene is changed from a G to a T, causing a premature stop signal to be sent to the cellular machinery that makes protein. You could also see the P dot written as P.GLU2143 star or P.GLU2143X. So you can see a star, an X, or a TER at the end of the P dot, and those all are indicating a nonsense variant. To reiterate, a nonsense variant could result in a shortened protein, but likely results in no protein being made. The last variant on this slide is a partial gene deletion that does not result in a frame shift. Here, a large portion of the gene is deleted, which is expected to cause a loss of functional protein. That's because a big chunk of the instructions to make the protein are missing. The first variant on this slide is a splice site variant. If you see a plus or minus with a 1 or 2, it indicates the variant is in a splice site. A splice site variant is a change in DNA that affects how it is processed into instructions for making protein. With the processing disrupted, the instructions are disrupted, and the protein can be made improperly or not at all. With the second variant, one nucleotide in the MED13L gene is duplicated, causing a frameshift variant. Frameshift variants result in the protein being made improperly, so too long or too short with many amino acids changed, or not at all. These are my references for the presentation. I hope you found it helpful in some way, and thank you for watching.